This is TWIS. This Week in Science, episode number 973, recorded on Wednesday, June 5th, 2025. Take a healthy science bath. In Bath? Hey everyone, I'm Dr. Kiki, and tonight on the show, we are going to fill your head with infections, solutions, and a big tiny fern. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. We live in an ever-changing world. Innovation is constantly reinventing society. It's why things that once were common are now rare, like phone booths, phone books, and the Rolodex, like typewriters, telegraphs, and carbon transfer paper. It's why your local hardware store doesn't carry a wide assortment of stone tools. It's why traveling from Europe to America takes hours and not months. It's why you see so few want ads for cart and buggy drivers. It's why fireplaces need self-starting logs. It's why CD players aren't options in cars. It's why nobody has a loose coin jar anymore. Change has been taking place since before you were born and will continue long after you hear these words. Knowing this, that change is inevitable. The question is, what kind of change do we want to see? And what sorts of change do we wish to avoid? And how long must we wait for another episode of This Week in Science? Well, it's coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki.
And a very good science to you too, Justin. And yes, we are back for one more. Yeah, we'll do it again. One more episode of This Week in Science. Here we go. Let's start this show. Thank you, everyone, for joining us. So glad that you're here for another discussion of the week's stories. I have stories about some space adventures because, you know, what is a week without going to space? A toxo-Trojan, water and life on Earth, thinking while menstruating, and the healthy Roman baths. What'd you bring, Justin?
Gene therapy curing deafness. Maybe a glimpse into the next pandemic.
No.
A little bit of H5N2 new news has been breaking. Cornell University study finding that, oddly, the color of a driver's skin seems to influence the rate at which they are pulled over in traffic stops in these cities.
Never heard of that before. Really?
I don't know. That seems like it's breaking news, right?
Yeah.
The giant, the largest
Breaking our society.
Largest species ever discovered on the planet is a fern. And what else? I had something else, but now I can't figure out what it was.
All right.
And good AI news.
We talk about good AI stuff all the time. It's
fun. But this one isn't hyped. This one's actually already. This is a good thing that's happening now already with AI out of Denmark.
All right, we are AIing ourselves into. No, it's just us. There's no AI involved in this show at all. This is all produced by us. We're doing there's probably some algorithms involved in certain aspects of the technology that are used, but we don't need to worry about that. Neither do you, because if you like this show, all we need is for you to make sure and show up every Wednesday, 8 p.m.-ish Pacific time for our live broadcasts on YouTube, Twitch, and Facebook. And you can be part of our chats that are live, the discussion. If you're a Patreon supporter, then you can join our Discord for chats. We also have a podcast because that's what the show broadcasting live is doing is gets turned into a podcast, which is then put out into all the podcast places. At least I try. And, sometimes there are places like Google Podcasts that they're like, I'm not going to do it anymore. And I think that I've got it in YouTube. And then YouTube's like, yeah, no, we don't like it. So I'm working out a few glitches there. But keep up
One
with
of the
us.
things,
Just subscribe. Look for This Week in Science website, twist.org, and just please.
we might be in the category of old newspaper to start fires with and Rolodexes, in that we are both a live show currently right here, right now.
Yes.
And there's no writers.
Right here, right
We don't have a writing
here,
team
right
that is behind us producing all of this material. This is just us talking. And so we are completely responsible for everything you hear.
Oh, wow. Great.
Not in the legal sense, but in the whatever other sense.
In all the senses. All of the senses. Oh, my goodness. I sense that it's time to start. Nope. I sense that it's time to start talking about our stories. Okay. Because I'm going to keep us on track. I have a son who has a couple of math tests this week, and I need to help him
real world
child needs to make sure he understands statistics, probability, and geometry before tomorrow and Friday. No biggie. It's great. Smart. Yeah. Okay. So what does statistics and probability and geometry have to do with my first story? Oh, because they are involved in the probability of certain companies like Boeing actually launching the engineers using geometry and physics and blah, 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 all that kind of, to actually launch a rocket carrying 2 astronauts into space.
So can I take a slight bite at that?
Why?
Boeing had nothing to do with the launch.
They built the Starliner.
Boeing is only the capsule at the top.
Yeah, the Starliner. Yes.
But the rocket is a different organization that was in charge of doing the launch.
Yes. The rocket has had issues. The Starliner capsule had some issues, but then it was ready and it did its tests very well. And so that's why the astronauts the last couple of times were waiting on the launch pad while they waited to find out that there were issues with the rocket. But in this particular effort, which is very exciting, the Boeing and rocket company and NASA partnered Starliner capsule actually left Earth's, terra firma. When it's in space now, it's heading for the International Space Station. We've got 2 more astronauts in space. It's very exciting. The Starliner capsule is created to be reusable, and when it comes back down in a few weeks, it's going to be landing on the ground and not out on a platform on the ocean like SpaceX's Falcons. It is going to be landing on cushioned balloons on the ground. So we'll see how that airbags. We'll see how that works. We're very excited about that. And I honestly, I wish these astronauts well. It's great to have more astronauts up in space. The more individuals that are up in space looking into the great beyond and back at our own planet, the more we are able to understand more about our place in this universe.
In terms of that re-entry, I think I would like the idea of parachutes, just because I feel like I understand how that's gonna play, right?
Yeah,
and about the old way though was to have them parachute into the ocean, which if I was the astronaut, that's the part I would have a problem with. Like, oh, are you gonna drop me in the middle of the ocean somewhere? I don't, that sounds like worse case scenario.
Yeah, I mean, we know from
this airbag thing.
Yeah, we know from NASA that ancient Mercury missions and Apollo missions. There was an astronaut who splashed down and was able to get out of the capsule, but was stuck in it. And if he hadn't been able to get out, he would have sunk to the bottom of the ocean and drowned. But he was able to get out, which is incredible. So,
Not
crash into the
ideal.
ground, crash into the ocean. There's always things. I mean, it's dangerous. This is a dangerous business. Joke from Kevin Unique in our Discord chat. Boeing built the capsule so if the booster blew up, the capsule would just bounce off the ground. Boeing, Boeing, Boeing. I just have to say thank you for that joke, Kevin. It just made me happy. Boing, boing, yes.
Also, we got Robert Barner in the chat room. Boeing, Boeing, gone.
Yes. But this capsule is also another part of NASA's efforts with various companies to be able to continue our efforts in getting to space. And the more companies they can partner with, that is to say, Boeing got a lot more money than SpaceX did for this effort. It's taken a lot longer to get here, but we'll see, how things move forward. And according to reports coming up in the future, we've also got Sierra Space's Dream Chaser is going to be headed toward the space station for a cargo delivery, and ULA is going to be testing its new Vulcan rocket, which is a successor to the Atlas V. So there's a lot going on in the space industry, the new space world, which is merging our public interests with private interests. But that's been the case all along, and that's why we've had certain missions maybe certain efforts last longer than they should have because of where parts are made and how things. Anyway, that's political pork. More political pork is that I guess apparently we're in a space race with China and India and Japan. And this week, China they did land their the Chang'e 6 lunar lander on the far side of the moon. And this is a probe that lifted off yesterday morning, 7:38 a.m. Beijing time, and it touched down on the moon, grabbed some bits of rock, dug into the soil, and what's in here? What's in here? And then they went, oh yeah, here's our flag, and they showed their flag about a little bit, and then it took off. And hopefully it will be successful, like its precursor, the Chang'e 5 probe, which went to the moon. The side we can see, the light side, the near side, not light side, the near side of the moon, and did the same thing, but this was on the far side. So they lose radio contact. It's a lot more complicated, and it's also very interesting because of the location that they have picked and where they're going to be bringing samples back from is this impact crater, which potentially is so deep that it may have some of the impact may have that created the crater might have ejected materials from below the surface, and they may be bringing back stuff that could tell us about the history of our planet and our solar system from more than 4 billion years ago. So it isn't just the far side of the moon; it's the South Pole Aitken Basin, and it's very, very exciting and interesting. Where I'm excited about it. Space, I think, is an international endeavor for humanity, even though we do have these petty borders, things that,
Well, yeah, I don't know. I think relying on, for instance, an authoritarian government system in Russia to launch things into space was a bad plan.
Yeah, whatever. Yeah.
There are differences. I will just state that not all of those things are petty.
It's not all, no. But so space, it's happening. It's very exciting. I think that good job, China. I'm excited. I want Japan to succeed. I want India to succeed. I want everyone to succeed and bring samples back. And if you want to put people on the moon, put people on the moon. Hopefully you'll bring them back too. I think this kind of adventuring exploration, these are historic moments, whether they are United States, China, Russia, anyone, but it's, the international community, I think, should be very excited for this effort that has is giving a boost to science if all the data is shared appropriately. Hopefully, open science, yay! So we can talk about it later. Whee! Oh no, I just closed all my stuff. Okay, you're up next. What do you want to talk about? More space?
Well, I'm not going to talk about Europe. I'm going to talk about China, where gene therapy that was designed to target a form of inherited deafness has restored hearing function in 5 children who were treated in both years. So this is sort of a follow-up. There was a previous version where they tried this gene therapy to affect 1 year, and it showed some success. But now they've done it in 5 children in both years, and these children experienced within several weeks post-injection better speech perception, gained the ability to localize and determine the position of sounds, were responding to their names, were dancing to music, and were repeating things that adults would say to them. So this is pretty stunning, pretty amazing.
Being able to repeat, that's amazing.
Yeah. This is the world's first clinical trial to administer gene therapy to both ears. And it is published. This is by the University in Shanghai, and the findings were published in Nature Medicine. So pretty amazing. There's some videos that go along with that too. So I recommend everybody search our website for the link to the story if you want to see those. The only thing is
what?
Well, I'm so jaded that I need to see this repeated in a, well, I'll call it Western world science situation.
Yeah, right. And we are in the middle of an understanding of where there's so many retractions from journals these days. It's the numbers of retractions are going up and up and up and up. And we've talked about that. And there is ongoing, just a report out this week again, that the retraction and rate is increasing. And so there are issues with publishing, right? So just put that out there.
And China specifically has been a major abuser for some reason of putting through scientific research that has a lot of questions about reproducibility. And so this has videos which may be very convincing, or you could look at them critically and say this actually is meaningless.
Right.
That you have videos. But if true, and I hate to have to even do that disqualifier, but if true, that is an amazing breakthrough. That because this isn't just restoring hearing to those who have lost it. This is basically introducing hearing to those who were born without, which I think is very significant. So this is okay. So we're showing the video, and here's a child, and they're speaking to the child like, "Hey, look over here," and the kid is paying no attention, no response. 6 weeks after the injection, "Hey, over here," and the kid immediately turns around and looks. And then I think this is the one maybe even where so you can see kids are dancing to music and having fun. Now they could have just had a happy child and, played some music behind it for the video. Like, again, my level of suspicion is only because I have read so many of these research papers out of China that just on the data that they provided in those papers didn't match what they were saying. And a lot of these sorts of things have been retracted again in the very recent future once they started looking in and getting really serious about vetting their publishing in scientific journals, which has become a very pronounced issue in the last few years.
Yeah.
So I remain suspicious because there's a real big push in China to be first in all of these things, and it's sponsored by an authoritarian government, which means people down the food chain of that decision making don't have very much say in what is being put out. That's a very important distinction. In the U.S., any researcher who does a study, even if it was paid for by a grant from the National Science Foundation, it looks at it and goes, you know what? That's not what my data found. Can say what their data found, and there's not going to be a pushback.
So this paper, you can look at it and yeah, this paper has been published in Nature Medicine. It is open access, so anybody can take a look at it. And this is why I think open access is pretty awesome.
Yeah.
This is where peer review isn't so much, peer tear down, but people and researchers specifically who are involved in this area of gene therapy with adeno-associated viruses, that they can take a look at the evidence themselves and really look at what is already a peer-reviewed journal article. It's not, it has been peer reviewed. This is Nature, but that doesn't mean there aren't, there aren't things that push through.
And to be fair, or not to be fair, to be unfair. How am I trying to say this?
Yeah.
I have some of those articles that I was talking about where there was an obvious conflict between data and what was being stated. We're in Nature, we're in Science, we're in JAMA. So they're not immune because of the prestige of.
They're not.
It
but
seems
open
as though
access
they've had a lower threshold for papers coming out of China than they have elsewhere, and it's a problem. And
I don't know that it's a lower threshold. That probably there's just a lot of publications, and so when you have a lot of publications, probability and statistics would show that if you have more publications coming from one place than another place, that they're statistically more probable to be accepted as a study or to get reviewed and get pushed on in the process because you have more of them. And so that is the whole science. This is the dirty nuts and bolts of science conversation. And, I think these are it's the dirty laundry that science as the institution is currently like sorting through and going like, oh man, that's the underwear. Those are the whites. Those are the this is my cold water wash, whatever. There's a whole bunch of reorganizing. Everyone has a different method, and maybe somebody or people within science can put the methods together in a better way. So that's what I'm looking forward to. Yes, we're not talking about it. I went on a tangent because I'm tired and I haven't slept enough, and we need to keep moving because it's already after 8:30.
So
hearing this is exciting. Where does it go now?
Yesterday. Well, actually, it's yesterday. Is it yesterday? I think what day is it where you are? It's yesterday where I am, but it's today where you are, I think. The World Health Organization confirmed the first human death from H5N2 bird flu. This is the first human case of H5N2 strain of the bird flu reported globally. And it's also the first time it's been found anywhere in Mexico. Now, this is a different strain than what we were previously talking about, what was previously reported as circulating in livestock in Texas, specifically cows, and affecting birds and barn cats.
Not just Texas, though. They found it. I mean, Texas is one of the places that you reported on, and how the veterinarians were responsible for really bringing it to the fore and discovering it. But they're finding it all over the place now, and slightly different mutations.
Yeah. So this is this is H5N1, which was what was in Texas that infected 3 dairy workers there. So it did that was making the jump to humans. This current patient is in Mexico. It was a 59-year-old with no history of exposure to any chickens or birds or other animals. On April 17th, so this was actually a while ago when it first happened, the patient developed fever, nausea, diarrhea, shortness of breath, was hospitalized 7 days later at the National Institute of Respiratory Disease in Mexico City and died that same day.
No.
So this is, well, we don't know. So they were very sick, and then 7 days later they died. So we don't know. That could have been a week before they developed the symptoms that strong. We don't know.
We don't know what the whole story is.
We don't know the whole story there yet. This is always the thing with these two. It's like, if it did kill them within a week, right?
Mm-hmm.
Actually, good news. and that it's harder to spread if it kills quickly. Initial cases die fast.
We've talked about that. Yeah.
And then they're not walking and spreading for as long. If you can incubate and have this for a month, you're going to encounter more humans if it spreads. Now, that's the thing. That's the other thing. They don't know how this person got it, which is not a great thing. That's not a great part of the mystery, but there is no. But what they were able to do, they did an investigation, and they could find no further cases were documented during the health officials' investigation of dozens of patient contacts through that period, and none of the healthcare workers have. So thus far, we have seen no example of either H5N2 or H5N1 versions of this bird flu demonstrating human-to-human transmission.
Exactly. And I think that is the big take-home point, is that we have not seen that yet.
Right.
This particular respiratory, as opposed to the Texas workers who had conjunctivitis, and FATA is asking if the workers in Texas survived, and yes, they have. They've been treated, and the flu antiviral medication has worked very well in treating those infections. So we've got, this is one of the things that I think is different in terms of, everyone goes, pandemic kind of thing. And we've been worried about bird flu because it could have a high mortality. It has historically, the different strains of it. The respiratory one is the one we get kind of more afraid of because it's respiratory droplets. It's getting into the air. We saw with COVID, masks reduced the prevalence of flu. So people who are sick, you can wear a mask, you can not go to work, you can go get your high temperature checked out at the doctor or a hospital, and they have flu tests. Like, that is a common thing. They test you like, oh, your respiratory stuff, we'll do like a pneumococcal pneumonia kind of thing, and we'll do like a flu test. We'll see, what virus it might be. We were doing that before COVID, and it could pick up if there were these the hemagglutinin and the nicotinamide subgroups. So there are certain aspects. It doesn't, it might not say this is bird flu, but it'll be like, okay, this is a flu. And so that knowing, and then you get people who are in areas that are rural or that might have significant spread of this kind of stuff. If there were person-to-person transmission, it'll get picked up. Even though we're not specifically testing for this right now, we do have systems in place. And so, like, at this point in time, risk for normal average person, very, very low. You're more likely to get COVID still.
Yeah. And also though, with that, and maybe COVID has been an exception for a while, the advice people usually get when they have these symptoms is to stay home and recover. Not to rush to the hospital to get it identified and then get sent home with the advice of getting plenty of hydration, drinking some orange juice, and staying in bed until you're better. So there's also part of that problem is that even though that testing system exists, the normal advice is to just stay home and recover. And it's not
so people
something.
might be, and if people are having mild infections, this particular person in Mexico had lots of other underlying conditions, he said. So it was a special case, right? And that's the people who are the people who are more likely to end up in the hospital for any kind of respiratory infection.
So because of this, let's see. Based on the available information, the WHO assesses the current risk to the general population posed by this virus to be low. This low risk assessment will only rise if it is determined that the low risk assessment is incorrect. There is no specific vaccine.
That's like a circular little tautological argument. Chasing my tail there.
It's kind of true. Like, it's basically what they're saying is like the risk assessment is low. If the facts on the ground change, it could be higher. And
we'll change as we go. And that is what observation. That's how it works. Yep.
Yeah. There are no specific vaccines for preventing H5 virus infections in humans. There are candidate vaccines that have been developed for pandemic preparedness purposes. But of course, without a outbreak, there's no real world testing of any of this. But like you said, there are antiviral medications. They're not vaccines, but it's medications after the fact that have shown success. So nothing to worry about unless that's your job, and then it's like the only thing you're thinking about
That's
right
the
now.
only thing you should be worried about. Yes.
That's the only thing you should be focused on.
And, there are very simple things that people can do if you work in places where the risk of any kind of infection, respiratory infection, are possible, or if you're going to be in a situation, saline sinus rinses have been shown to be really good at rinsing stuff out of your, get rid of it. It helps your allergies. It helps you not get sick from viruses. It just rinses stuff out if stuff's not allowed to get attached to all the cells in the membranes. Washing your hands, making sure that if you are sniffly yourself, wear a mask so you aren't going to be propelling things into other people's air.
That's after
there's all sorts of there's normal things.
Avoid
everybody.
raw milk. Avoid raw milk because they're finding virus in the milk.
Yes.
In the raw milk. Okay. Pasteurized.
Yes.
Fine. Don't kiss cows. Avoid licking chickens.
Which is so funny because last week there's a Danish, I think it was from Denmark, a study on what was it? The cow hugging, Kuverhugen or something like Kuverklugen or something. There's something called cow hugging, basically, where people go to the country to be with nature and hang out with cows to feel better. And so, with this in mind, maybe, don't hug them. I mean, look at them from the fence. Yes.
And the last point is, this is, again, the first time H5N2 has been seen in humans. The fact that it's not a respiratory transmission between human and human is only going to be that barrier as long as more humans aren't getting it. Because the way this thing is going to mutate, the way it's going to find a host and find a new way to survive is by getting into humans long enough to become altered in a way that it takes advantage of the new surroundings. And so it's very critical to prevent this in the animal population as best we can.
Which is hard to do in the first place, but minimizing the spread. Now that we know there's stuff going, we know it's constantly playing at the edges. Can I get in? Can I get in? What am I going to do? I'm a virus. I'm changing forms. I'm trying all sorts of things.
We've been talking, the bird flu has been trying to get in for a long time. The way you put it there is it hasn't given up. It's
constantly poking. What can I do? How can I get in? Oh, now it's Kernuffeln. You have to tell me how to say it. Yeah. Anyway, cow cuddling. Don't do the cow cuddling, everybody. I like.
Cow is cool.
Kernuffeln. Cow cuddling. Anyway, okay, let's move on. I did have some more stories. Justin, you do love the Toxoplasma gondii.
How did you find a toxo story? And I have not seen this.
Dun, dun, dun.
I have no idea what this is.
Yes. Well, some researchers at Donnelly Center, I shouldn't put that accent in it because it's from Toronto, Canada, University of Toronto. This is the Donnelly Center for Cellular and Biomolecular Research. They have published a study in Oxford Academic Journal's Virus Evolution called A Parasite Odyssey, an RNA virus concealed in Toxoplasma gondii. The researchers were interested in what might be part of making certain strains of Toxoplasma gondii more harmful in their infections, the parasite. How does toxo get in? Why are some strains more likely to cause blindness and other strains more likely to cause, severe inflammation? What's going on there? And so these researchers at the University of Toronto, they did a scan of RNA because RNA is actually more of a signature of things that are working and things that are at work within a cell, as opposed to stuff that's just kind of there, you know, not, just it's there. It could be a dead, it could be whatever, could be doing a job or not. But RNA is it's a signature of things happening. And yeah, lots of RNA viruses like to stick themselves into parasites, into human cells. It's retroviruses, it's RNA. It's great, wonderful. We love RNA. Could have started everything. And now they looked at a big neuronal data screen at RNA, and they discovered there was over 150,000 RNA viruses they looked at, and what they used an open science platform called Ceratus. And in their analyses of toxoplasma infected neurons, they discovered that there are some very specific RNA viruses that are present inside the T. gondii parasites. So the two of them that are specifically known to be really, really bad, French Guinea has a T. gondii parasite strain called Rub. And then in British Columbia, there's a strain called Cougar. And anyway, French Guinea's Rub causes organ failure and fever. And in British Columbia, the Cougar causes infectious blindness through ocular toxoplasmosis. But they're bad. And so why are they so bad? And they pretty much said, well, oh, wow, we got 19 RNA viruses, and they seem to be really strong biomarkers. So if you are looking to find out if somebody is infected with Toxoplasma gondii, you can actually use these RNA viruses as good markers of that infection, where you won't necessarily get the actual T. gondii parasitic load or what's happening within the nervous system. So the RNA can potentially be used now, and there are some of these viruses that they think not just can be markers, but are probably working in a synergistic manner to make the body's reaction to toxoplasma hyperactivated. So it's the virus and the toxo that make the results so much worse. So, toxo, it's great on its own, but it's like unwrapping a Kinder egg. You got more on the inside.
Yeah, it's such an incredible thing then, because the toxo, and this is the example in cats, reproduces, and then it has to get pooped out, right? Yes. As the parasite again. And most other mammal species, almost all, it can't reproduce. But the little cells that would become the gamete cells or whatever, they're small enough that they can travel out of the digestive system to different parts of the body. And if those are containing the viruses, oh my gosh, that's a delivery system, right?
Yeah.
That's just a delivery system to the brain, to different organs, and then the virus that had no access to these systems otherwise, is past all these human defenses and can go to work. And it may be why there's this phenomenon of toxo-infection being able to be dormant.
Right. Versus
for a very long period of time.
Yeah.
Separate from direct exposure, and then have the effects, which is, I don't think has been an understood part of this infection, is how that's even taking place. And if there's, what do you say, 19 viruses that they discovered in these parasitic gametes? Oh my goodness.
Yes. Yeah, so there is there's a large number of, it's like 19 RNA viruses that they could use as possible markers of the infections, not just in people, but in other animals as well. And so then you don't just treat the virus, you go, oh, this virus, it goes with toxo, so now we treat the parasite. And so it
And
could
then the
be,
parasite's almost like not even a thing at that point. Then now if the effects are the virus, then you just need the antiviral that can affect those specific. But on RNA virus, oh gosh, it's deep inside the brain or the organs. It's like very wow.
Mhm.
My goodness, that's a pretty huge breakthrough, though.
It's
That's an amazing
duplicitous,
story.
isn't it? It's like
It could answer
doubling up.
so many of the last decade's questions about how Toxoplasma gondii was affecting people.
And why different, like you said, why are different strains? I mean, different strains,
Mhm.
they're going to act differently. The body's going to respond to them differently. But why is there such a huge difference
Yeah,
between
and it's
certain ways that it acts in people? Yeah.
yeah, there was exactly. Like the aggressiveness
Mhm.
in different areas is so unbalanced to others that it's been a, and then people are like, well, maybe it's a cultural thing. It's done like maybe it's a human user end of cat user end problem or things like this, like trying to figure out how these things develop. But yeah, wow, that's
Yeah.
huge.
So yeah, I think it's huge. I think it's very exciting. I think this opens up a whole I don't know, it's a whole new world to look at parasites and how they interact with people. It's a whole new view on how everything's just a vehicle for RNA to do what it wants. And this is my musical. No, okay, I'm done. Do you want to talk about something else?
Well, I can. With all the doom and gloom news surrounding AI, not to mention the relentless hype machine that's trying to drive stock prices higher, there is good news in the AI field coming out of Denmark. Using artificial intelligence, breast radiologists in Denmark have improved breast cancer screening performance and reduced the rate of false positive findings. These are results published in Radiology, the Journal of Radiological Society of North America. So basically what they did was they compared a group of women aged 50 to 69 who underwent biennial mammography screening in the capital region of Denmark, which that's right outside my door. In the first group, 2 radiologists read the mammograms of women screened between October 2020 and November 2021 before the AI was available. And the screening of the second group of women was performed between 2021 and 2022 with the assistance of AI. Now, the trick to this is there's also a couple of things. When humans do these readings, they have a second read because they're trying to avoid false positives. So humans have to read everything twice. These were first pass runs by the AI. And, if half of your population just about is going in for these readings, there's only so many radiologists who can read through results. And so there's always delays and a rush and a just unbelievable amount of workload to do things once, let alone do them a second time.
Multiple times. Yeah.
And then those that get identified as potential have to go then to a specialist a radiologist who will also look over them basically a third time. So what they did was the AI was sending off to the specialist after for a second read, but was doing a first read by itself. The results were pretty amazing. They had in total 60,750 women were screened without AI. 58,246 women were screened with the AI. Compared to screening without AI, screening with the AI system detected significantly more breast cancers, almost 12 to 15% more, and had fewer false positive rates, dropping that by about 30-40%. So, I mean, also then, and it reduced the number of recall rates by 20%, which is somebody they've looked at your results and they want to do the testing again. And so you have to come back in.
Yeah.
They say that the reading workload was lowered by 33.4% for the specialists. The positive predictive value of AI screening was also greater than that of screening without AI. And they had a higher proportion of detecting invasive cancers detected that were 1 centimeter or less in size. So they were also better at detecting the early ones. And according to this, there's also room for improvement because radiologists, when they're doing the read and the second read version, they typically have access to all of the previous screenings. The AI system currently only had what was placed in front of it for that specific screening. It had no historical information to add to its calculation or its analysis.
But it's a model, so it probably had some amount of learning data before it's got.
Of course, general,
so it's general probabilistic association.
But if you can imagine adding the additional information to the detection, showing any sort of trending or lack of trending, that could potentially increase its detection rate and lower false positive rates as well. So, for instance, if there had been something that was identified by a radiologist that they had biopsied even and found benign,
right?
The AI wouldn't know that. Right? So
it would compare. Yeah.
So by filling in more information, feeding it to the AI, greater and greater detection. So that to me is like one of the definitive stories of what AI needs to be used for, as opposed to just focused on replacing all of the workforce.
And according to another study out about AI, basically, if you tell people that, AI could take your job, then people are like, well, why do I want this job to begin with? And people are like, maybe I'll quit this job. And then suddenly, like, nobody wants to work anymore. And they're like, I'm going to go get another job. And then you're like, oh, I guess we got to get the AIs. Why does nobody want to work? Oh, my gosh. Yeah.
Yeah. I have a friend who will go unnamed who quit. They're a linguist, and they were working for one of the bigs tech companies, and at some point realized that their job was designed to get rid of the necessity of them. Exactly kind of what you're saying. It's like they're training this to do my job.
So
okay, goodbye.
Bye.
No, I don't need to help. I don't need to help you with this. Goodbye.
Yeah. I don't need your stinking captcha. What do you mean?
Take this gerb and serve it.
Oh, serve it. Let's serve it. Yeah, let's serve it. Okay. Really quick 2 stories that I wanted to throw together that I saw that I thought were very interesting this week before we head to the final part of our show. This week, both papers published in Nature, researchers were looking at life, water, crystals on our planet, and not in the new agey kind of way either.
Then never mind.
Yeah, no. So in Nature Geoscience, just June 3rd, published a couple of days ago, researchers publishing on 4 billion-year-old zircon crystals that were collected from Western Australia's Jack Hills, which are known to be some of the oldest rock formations on Earth. How does that happen on a very volcanic, active planet? Well, it happens when there's a particular part of the planet that doesn't get sucked down under the crust into the mantle or isn't being shoved out. So it's a very stable area at this point in time, and it hasn't been eroded away. And it's still above water, which makes it great for us to be able to look at. And in this particular study, the reason they looked at zircon is that zircon is a crystal that has different forms of heavy oxygen isotopes. And they were able to look at oxygen 16, which can evaporate a little bit faster than oxygen 18, which is a bit heavier. So these isotopes are like just slightly different conformations that have, and so they interact with the environment just a little bit differently, but they're still oxygen. So they can get trapped in water. They can become part of crystals, all sorts of, they're still just oxygen. It's just a slightly different molecular makeup. And so these researchers like, all right, so we got this really old land, we got these really old crystals, let's take a look at them. And, we know that oxygen 16 can evaporate a lot, oxygen 18 doesn't. So if we look at the ratio of these things, well, we're going to find out fresh water should, because it has more water in it than or salty water has more salts and other solutes and other things that'll hold the water together. So over time, you're going to have fresh water that's going to be concentrated in oxygen 16. At least that's the way that, because that rainwater goes through the ground, it's going to go through down rivers and streams, and it's going to then get pulled into the ground, and it'll end up interacting with rocks like the zircon crystals. And so anyway, they were like, let's look at these crystals that we dug out of a really old rock. And they looked at these really old oxygen 16 and oxygen 18 ratios. And they determined that based on their measurements, that the rocks suggest that there was fresh water on rocks that were had come up out of the salty water. And so that there was a freshwater cycle happening at least 3.4 billion years ago, which correlates with some of the oldest evidence for life on Earth, also found in the Pilbara region of Australia. And additionally, they suggest that it may also go back to maybe up to 4 billion years ago, based on what they're looking at and the way that the data works out. So their question is, okay, old zircon rocks, were they out of the water, and was it like a cool little spot where there was situation that allowed life to do life on the surface of the planet and not in a saltier, brinier place. So I think the putting together the timelines of like the 3.4, 3.5 billion year old evidence of life, which is still questioned because, it's pulling rocks apart and looking at molecular conformations as opposed to actually having a fossil. But this kind of evidence for freshwater cycles on the surface of the planet at 3.4, going back potentially to 4 billion years ago, suggests that window for life evolving out of the oceans on the surface of the planet is very likely.
This is the
yes, but
the life signs. No, not a yes, yeah. But yes and. The life signs that we had seen that you were mentioning, there, these things that look like they may have been caused by a biological bacterial life form on the surface of a rock.
Yes, maybe, yes.
That are the 3.4 billion. That was thought to be under saltwater.
Some of it. So it is very possible, however, that this could have been a hydrological cycle that allowed for life to chemically react, like for the origins to react on the surface of the planet, that it was a spot that maybe it would have been warm, but there would have been rain and there would have been anyway, go ahead. Yes.
Oh, I mean, but isn't this though then, like, this is the first evidence of freshwater, the furthest back evidence.
Furthest back evidence of fresh water as opposed to salty water.
Yeah. Cool.
And that has been a big question. People have wondered, how early did freshwater really appear on the surface? And these rocks suggest it was a long time ago. And they looked at the rocks. They also did computer simulations, and it was only the simulations that involved freshwater that recreated the results that they got from the rocks they collected. So anyway, fresh water could have started on the planet a long, long time ago. But, 3.4 billion years, probably 4 billion years ago, they need more evidence. Still need to have like a lot of, there needs to be better evidence. And they say themselves, they don't have that evidence. Moving on from there, though, it's like, what are the evidences of life? And when you're looking at rocks, fossils are usually the hard stuff that's left over from stuff that was living, or it's, chemical in some nature. Like we are now looking at these evidences of microbial mats and biological bacterial metabolism. But larger organisms, especially early organisms that didn't have skeletons, didn't have scales, didn't have all that hard stuff, they get lost. And so there's been a big question as to how long ago things like sponges, the earliest ancestors of all of the, big vibrant life on the planet that we like to talk about, it might have gotten their start. And so we can date.
Not a ton of evidence for Precambrian explosion. We know like life was exploding into all these diverse forms, but whence they came, because this is like the first like exoskeletons, and so like you say, fossil evidence that we have.
Mm-hmm.
Explosion around the world, but before we call it an explosion, that doesn't mean that life.
It wasn't that
first
fast.
Exploded. Then that's exoskeleton explosion, right? That's a fossilized evidence explosion is what took place. Before that, we know there must have been tremendous diversity of life. You just can't see it.
You can't see it. You can't, and we have to find other ways to look at it. However, there are certain situations where it's just the right conditions and soft-bodied things have been preserved. And there is an area in the Dengying Formation of South China that has been dated around 551 to 539 million years ago. And the researchers have placed it as an early sponge fossil. No, but it's not a sponge. It doesn't have the hard spikiness and the crystallization of the structures that are the what define more like our early ancestors more and to even more modern what they call biomineralizing organisms. And they suggest that there may even have been a divergence of sponges that biomineralized with silicas, and then others that did not and don't have like glassy, spiky bits in them. And so this particular species did not have those, what they call biomineralized spicules in it. But they did find a really cool impression, and it's not like a comedic impression, but they found an impression of this sponge species in a rock. And one researcher was like, hey, look what I saw in the rocks. And they sent the picture to another researcher, and that researcher's like, there's really interesting looking lattice work in the rocks. Why would the lattice look like that? I don't know what's happening there. And so they started investigating more closely this impression that almost looks, I don't know, like a tweed or, some kind of nice, checker fabric weave. Yeah. It's a very nice like basket weave. I don't know. There's some major axes, minor axes for cross structural formation. And so it's a very interesting looking specimen, and they got really up close and personal with it. And in getting up close and personal with this specimen, they were able to determine that it's really old, and it's really not normal that it would have been preserved in this way. And so the situation in this particular region of China where it was found suggests that it is a very old offshoot of our early ancestors, and suggests that we should be looking at other signals for, how do you find the old evidence of life on the planet? And we got an idea that, with sponges, it should have to be this biomineralization, but maybe not. Maybe there are other avenues and different traits that should be searched for in different places. Yeah. Sponges, not just for your kitchen sink, huh? No, they're old. Gosh. People should not let me not sleep enough. It's just... This is This Week in Science. If you want to have a good nightly rest, if you want me to have a good nightly rest, make sure you tell your friends about TWIS. I will sleep well knowing that you have shared This Week in Science with all the people that you know, gotten them to subscribe to us either on our YouTube, Twitch, or Facebook channels, or on any of those podcast places where we show up usually. And, if even better, man, I'm going to sleep and dwell in that dream world of bliss, knowing that you're supporting TWIS by either sharing us with your friends or other people in your community, or going on to Reddit, to the podcast subreddit, and going, hey, I can answer this particular query that somebody made about podcasts to listen to on long drives, and tell them about This Week in Science. Maybe that was a particular call to action, but I think you all ready.
It's very specific.
It's very specific. It's the podcast subreddit. Go there, please. We need more twists in there. I love Ali Ward and ologies, but come on, people. Let's get back to the roots of podcasting. Let's get some other people in the lists. Moving on from there, though, we also have our Patreon. If you are able to subscribe and support us through Patreon, $10 and more a month, we'll thank you by name on the show. There are also lots of fun little gifts that will be sent to you through Patreon at various levels of support. I really, really appreciate everything that you all do, all of you in the chat room, in the Discord, everywhere. Thank you for being here. Thank you for your support. Can't do it without you. All right, Justin, let's come on back. It is time to talk about something uplifting and wonderful.
Yeah, so this is perspective, perhaps. We were just talking about old sponges and tiny viruses, all sorts of things that come up on the show. This is the largest life form on planet Earth ever discovered, and it was just discovered.
Largest.
It is by far the largest. It is 50 times larger than a human.
What? So it's what are you talking about?
It is larger than any other mammal animal species on the planet.
An aspen grove.
Nope.
Or an underground fungus.
Nope.
Truffles.
Bigger still. Still bigger.
Okay.
Still bigger. And it's actually a small fern species that I can't pronounce, but I'm going to give it a shot anyway in real time. Temestipteris oblanceolata. Oblanceolata, maybe.
That's great. Yes, it's wonderful.
It's a little fern species that is found on the island nation of New Caledonia, which is a little island about 750 miles east of Australia. The species genus is an understudied group of plants that consists of about 15 species, mostly found across the range of Pacific Islands and Oceania.
It's a cute little fern too.
It's a
Pacific Northwest. We have Pacific Northwest. We've got all sorts of big ferns. This is a cute little fern.
It's a cute little fern with a genome 50 times larger than human DNA. Genome.
Why?
50 times larger.
Why?
It is now the largest known genome of anything on the planet. The previous largest was a species of Japanese flowering plant. So it was also a plant that was the largest genome previously. But it is now this New Caledonian fern. Now, you might think, hey, whoa, what is it? Why? I don't know. This one just seems to pick up through evolution new genomic recipe material, new genomic ingredients list, and hangs on to them. But it doesn't necessarily make it more powerful or strong. There is something to be said for picking up new DNA and getting rid of, sort of shedding the old clothes, getting rid of some of the old ingredients, the old tools. These ferns tend to actually not be the best survivors. They're surviving on these islands, which is great, but they may be very, they have a hard time with photosynthesis. Climate change might affect them. Soil changes might affect them pretty quick. So it's not completely understood what all this extra 50 times more than it would take to make a human DNA is doing in the genome of this tiny fern. But it's not a super plant by any means of like invasiveness or survivability or robustness. That hasn't taken place. But my goodness, what an interesting plant to study then. Because my guess is, because of where it's located, it probably did not maybe start on this little island in the middle Pacific. It probably has undergone adaptations from various other locations over how long, I wonder? How far back has it been collecting these? What can the adaptations that are in the genome but perhaps not being used currently by this little fern, what can that tell us about its history, about its path of evolution into this modern world? It may have a lot of ancestral knowledge, or it just might be like a really rapid adapter and these might be changing all the time. It needs to be studied more.
Well, yeah, I mean, how much is wrapped up in histones that doesn't get expressed? How much is actually, like the part that's used versus the stuff that's just like, just sitting there in the recycle bin, but didn't actually go out for recycling, like your computer app where it's like, I thought that I threw that away, but you didn't because the recycle app didn't like actually do its job. So suddenly you have no storage, except this has plenty of storage. It keeps adding more in its tiny little frame.
It also makes me wonder, like, would this be a very useful plant for like biotechnology, or would this be a very unuseful thing? Because with so much going on in terms of the size of the genome, would that make it easier to manipulate to do something, or much more difficult because there's all this other competition with what could be getting turned on and turned off? I don't know. But
I think it would be an interesting comparative species, right? So you're tiny, you have a big genome. What are the genes that are necessary to make you work? And what are the things that probably could have contributed to the collection of the stuff that doesn't? Or do you use all of those genes? Are they all just slightly expressed at some point? I think that these directions of research are going to give us some interesting perspective into evolution, into different evolutionary paths and mechanisms, adaptation. And especially if this is a plant that will not be able to survive climate change and if you've got that big genome and you can't use it. I don't know.
More human-y news.
Okay, good. No.
A Cornell University study has found that the color of a driver's skin seems to influence the rate at which they are pulled over in traffic stops in the city of Chicago. Black drivers, it turns out, in Chicago were significantly more likely than white drivers to be stopped by the police, regardless of where in the city the stops took place or other demographics related to the driver. The study is basically a confirmation that racial bias in traffic stops is taking place and is basically a form of institutionalized governmental discrimination, and so should be addressed. In addition to police stops, these researchers were investigating ticketing by speed cameras that are deployed across the city. And they find that, the black drivers were ticketed roughly in proportion to the racial makeup of drivers in those locations, which makes sense. On streets where data showed half of the drivers were black, and about half of they had about 50% probability of being issued a camera ticket. In comparison, police, which is human, initiated traffic stops showed a strong overrepresentation of black motorists relative to the proportion on the roads. And it showed the reverse for Caucasian drivers. On a street with approximately 50% black drivers, they comprised about 70% of police stops on average. On a street with 50% white drivers, fewer than 20% of the stops involved white drivers. This is a quoting from Wenfei Zhu, assistant professor of city and regional planning in the College of Architecture, Art and Planning, the director of the Urban Data Research Lab. This is the first time that we've had such a granular block by block understanding of a bias in traffic enforcement, showing that police disproportionately stop black drivers at a higher rate. So they had used a bunch of cell phone capturing data, as well as looking through, they did Freedom of Information requests to get Chicago Police Department data in 2019 on nearly 650,000 traffic stops and more than 700,000 tickets that were issued by the roughly 160 traffic cameras across the city. 700,000 tickets from 160 cameras? Wow. They compared those demographics. Yeah. That's a lot.
It's a lot of tickets.
From not very many cameras. Like so few cameras are doing that many tickets. My goodness. The scholars compared the demographics from events on the road, the racial composition. Basically, regardless, this is according to again, regardless of where you are in the city and the makeup of drivers on the road, black drivers are always a higher proportion of those that are stopped.
Yep.
So if anybody was thinking, oh, I know there's racism, but how do you prove it?
Yeah,
well, with data. And here's the data. And it's proving racism. Like if you just don't believe people who've been telling you these stories forever, here's data that Chicago police are racially profiling in traffic stops.
And I think that's the big point here is, the question is, okay, how is this surprising? It's not surprising. People have been talking about this for a long time. This is very well known as an anecdotal trend, as an experiential trend for certain, for some people, right?
But to be fair,
what's surprising to me is this is the first time that there's been a real data-based study to actually show it. Like, to me, that is surprising.
It's not surprising?
That's amazing. What? Yeah.
That the police department didn't do this study to find out if they were racial.
If they were, yeah.
That's what's not at all startling. And that's why it is biased, and that's why there's likely racism involved, because the organization that's doing the racial profiling should have been the first one to recognize, hey, look at our own data.
Look, wait, they're not self-policing?
Okay, so driving while black is a known thing. However, and I will say this is a good argument about driving while black, is that if your experience is of those around you is somewhat segregated, you might say, well, I know plenty of non-black people who've been pulled over too. And if you're only surrounded in a predominantly black neighborhood, pretty much everybody around you in your neighborhood who's gotten pulled over would also be black. So it would seem like targeting. And so you could make the arguments that, it's just perspective, or people are too siloed and what they're counting is their data. You could make all those arguments. Gone now, because now we have data. We have data that shows, even if you believed or thought you knew one way or the other, the data actually shows institutional racism and racial profiling is taking place throughout the city of Chicago, regardless of the demographics of other than racial, regardless of the demographics of who's being pulled over.
And this is the kind of study that we just need more of. We need to we need to see this in every city. I want the data. There are traffic cameras all over this country. There are you can do Freedom of Information Act, or just go to the police and say, I want to look at your data for who you're pulling over. Do these correlations. Let's see what's happening in different places and how it's working. I want to see how it breaks down across the country. I want to see where, I want to see it. And this is what we need. And this is like a great model that I hope that is repeated over and over and over again.
And here's the other thing I just want to say on this too. Like, I kind of like don't like the idea of like sneaky traffic cameras. They should have like postings like, hey, we have traffic cameras in the area. Because I think if you police driving to the level of having a camera on every block, you'll get a ticket on every other block. Like, something will happen. Okay. Yeah. But on the other hand, now this is the greatest argument for traffic cameras I've ever heard of, which is, why do you have police pulling people over at all? If you can get more tickets with the traffic cameras, with fewer of them out there, and they're not racial profiling people, that can also, because also these traffic stops, these 650,000 traffic stops by police are not all ticketed. There's a lot of the pullovers where they
don't end up in tickets. Yeah.
They're asking questions or wanting to search a vehicle or finding out who the person is that have nothing to do with giving a ticket. So why do we need the police to write tickets if we can do this with cameras? We could actually get the racism out of the system with the machine, even though we were maybe getting ticketed more. So we have to decide. We have to decide.
What do we want?
How do we fix it? Do we want? Do we want the robot overlords policing us? Or do we want the racist cops policing us? And I kind of lean into the robot overlords, to be honest with you.
It's not a robot. It's not even a robot. It's a light sensor. It's basically like, did you cross the line after a certain signal? I mean, this is like, that's basic.
That's a robot. That's a robot. Not
it's a basic.
AI.
It's a basic system. Yeah, this is input,
because
output.
AI is still going to be racist, because AI so far in policing and judicial systems has also been racist. So that we can't go there yet. But the robotic or automatic, I guess that would be, maybe you would like that term better. Automatic ticketing.
Automatic ticketing. Okay. Yes, the system there. But this story has now brought me. Made me aware of the Urban Data Research Lab, which looks to be doing this incredible research and looking at what they say, measuring neighborhood dynamics with big data, looking at legacies of housing and real estate in the 20th century. They have projects based on urban renewal, segregation, redlining, looking at ghost cities in China, walks in the park, digital metatis. There are some really they have courses you can take. They've got a large number of papers. This seems to be a very interesting organization. So
we should probably reach out to them to do an interview at some point. It's one of the things. It's one of the things I really. One of the things I really appreciate about living in Denmark, or have appreciated. Not that is that there is a very aggressive governmental housing authority.
Yeah.
That it's sort of like reverse redlining. If they see that a community has become too disproportionately impoverished or racially segregated, they will basically force the housing to be redone and for people or things to be moved. They aggressively pursue disproportionate areas that are becoming underserved economically by changing the demographics. Sometimes it can be like if there is what you would call, there's not really like slums in Denmark, but if there's like a neighborhood where the housing values are very low and because people are very poor, they'll say, okay, we're going to tear it down and build all new stuff and then offer people other places to find living or, Anyway, it prevents exactly what it looked like. Some of those infographics from that Urban Research Center were showing. Where we isolate people from opportunity and increase the likelihood of all the problems that we see.
Yeah, of tribal dynamics. Let's increase the disparities between different groups in our. Let's keep. No, okay.
It's never worked.
It's never bad ideas, everybody. All right. I'm going to start out this last little couple of stories of the show with a talk about, what it's like to be somebody who menstruates. You go through.
Are you saying that word right, or am I just hearing it wrong, or did I just never know how
menstruate?
Menstruate? Is that what it is?
Menstruation. Menstruate.
It's not just menstruate. It's
menstruation. Menstruation.
Okay.
The cycle.
I don't
period.
Know. We know what we're talking
there
about.
There we go. We're gonna flow through this. Anyway, researchers have known for a while that there's a lot of evidence that women perform better spatially, have less chance of being injured, or actually they have a higher chance of recovering from injury during different phases of the menstrual cycle. And so the question is, if the physical stuff has been shown, what about cognitive? And if you are somebody who menstruates or know people who menstruate, then you may know that there's, sometimes the most, I'm so cranky. Gosh, I can't remember anything. Oh my gosh. Oh, I'm tongue-tied. I can't speak. I mean, I swear, if you were to go back through all of the TWIS episodes, historically, you would probably see a scale of my ability to run through the Patreon supporters at the end of the show without with fewer or more tongue-tied mess ups or word.
You're trying to
word thingies.
What I'm hearing, if I can mansplain for a moment,
don't you?
What you're saying is that menstruation brain is basically equivalent to normal men think.
Man think, but it's changing.
Normal man brain.
It's changing throughout the month, as opposed to it's just during the day. Like. People who do not menstruate, they are typically on just like a more like just during the day kind of cycle. But this is it. I don't know. There are definitely differences, but it depends on whether you're on contraception that keeps you from menstruating, whether there's hormonal control, whether what's happening in this. But for those who go through a 21 to 28 day hormonal cycle, there are things that we can sit down and talk about and go, oh yeah, that's totally this week. This is the week where I can get things done. Oh, that week, that's the week where I want to lie in bed and I can't do anything. Oh, this is, and then there's all sorts of things. And there's like the middle schmerz, which is, why do I have to have that? But anyway, researchers decided that they were going to not just look at the physical influences of menstruation on the actions and behavior of individuals. They the University College London Institute of Sport, Exercise and Health has published in the journal Neuropsychologia an analysis of 241 participants, 96 were male, 47 not menstruating because of contraceptives. So there were comparatives within the data set. They did a bunch of cognitive tests 2 weeks apart. So the groups were all split up, and every 2 weeks you'd come in and do a different cognitive test. And sometimes the cognitive test was like, hey, you should, I don't know, react to Winky. I- seriously, they had, like, I'm going to show you this picture. There's a little smiley face with the dude Winky, that they're like, hey, the stimuli, respond if Winky's winking. Do you see Winky? Hey, don't do anything if Winky's not, whatever. Anyway, different response trials. So, like, a response speed, which is attention. What do you see? How do you process it? Are you paying attention to it? And can you react to it? So these are cognitive, sustained attention based tasks. There were other tasks involved that were more related to pressing a space bar in reaction. So it's, again, reaction time, reactivity, attention. And this is a very simple task that has been used for studies through the years. Okay, people, how fast can you respond? This is, I think the Exploratorium in San Francisco, when I was growing up, had press the accelerator when the light turns green kind of a test at one point in time, which is a very similar measure of how attention works. Additionally, the other tests were rotational and cognitively challenging in terms of being able to geometrically determine the size, the shape, the orientation of a cube-based Tetris-like structure. How many cubes are there? Are the cubes the same amount as the other cube that you saw? Does everything work together? And how long does it take you to work through that problem? I don't know. Some people do it faster than others. So there were a number of these different tests in which they measured all of the people. And this one, I think, again, is like related to determining kind of mental physics. You're supposed to press a space bar when you think that two balls are going to collide. So they're both leaving different sides of the screen that the people are looking at various moments, and press the space bar when you think they're going to collide. So again, reaction, but also cognitive processing of stimuli, visual stimuli mostly. A lot of this is stimula-based stuff. Live in the world. Is this what it really means to be cognitively active in the world? Your brain working, remembering how to say things? I don't know. But the researchers did find that there were no differences between men and women generally throughout the entire study. No, or menstruating people and non-menstruating people, I will say. No differences. Everybody fell within the same error bars. They're all looking good. But there were differences within individuals. So within the individuals who are menstruating, they performed differently. And it didn't matter which cognitive test it was. They performed differently at different times of the month. And it's the week of the menses, of the menstruation, when those people were actually at their cognitive finest. And this is interesting because that's the week that the people were like, I'm not doing so great. I don't feel so good. Nope, not really. So there was a discrepancy between how they self-reported, how they felt, but then how they actually performed over time. And so that in itself is very interesting. But, the overall data is like, okay, overall, there's no difference between anybody who's performing on these tests. Like, basically, they're all falling in the same statistical probability region. I got to talk to my child about this before bedtime. So that's what I think is really fascinating, that there is a self-understanding of how you feel and you might feel physically bad. You might feel like you're physically not, or, or mentally not doing well, but the actual performance was better. And so the question is that performance better because you're overcompensating for the fact that you're not feeling great? Or is it that that actually is a period of time when whatever the hormones and the things are doing, they are actually making you do a little bit better? Because maybe, oh, you need to survive right now. and, and this is maybe dangerous for you and predators at this moment in time, if we go back to an evolutionary kind of standpoint on it. You're not going to say anything in a mansplain.
Well, I
Okay, here we go.
normally I jump into everything, and this one I'm like, well, I can't really give you any kind of anecdotal example of what it's like to have menstruation or menstruation brain. I don't have a thing for that.
No, you wouldn't.
Although
and
I
you,
can
you
tell you,
shouldn't necessarily. You can talk to people about it. But
I can say that sometimes when I'm feeling very stressed or tired, or I will say even a little bit of, to the point where I'm like annoyed with everything. Sometimes my critical brain faculties are a little bit like lasery focused.
Yes.
Right. Like there's something where I'm like,
Yep.
I'm not taking anything casually right now because I can't. I'm too tired to not just either completely concentrate on this or irritated to get through something real quick. That all of a sudden it's like all, but I don't know if that has any relation in any way to anything that we're talking about here, because I wouldn't know.
And I think, that's the question, that I was bringing up is like, what are the factors that are, cycle? There are the mechanistic factors that lead to the behavioral output, right? So there's stuff happening underneath the hood that influences the way you feel, and then that influences the way you act, and like all these things tie in together. How does input come in? How do your systems respond to it? And like you said, if you are in a space where you are tired, or you need to get things done, or there's whatever it is, that laser focus, you're not going to put up with stuff. Regardless, I found this and find this a refreshing study, because it's something that needs to be looked at, because there are real changes that occur throughout people's lives for everyone who menstruates and not. But the people who menstruate, they've got ongoing monthly changes, and then comes the end of menstruation. And what does that mean? And that's just a whole other thing. So this is a social, public health, mental health issue that needs to be taken very seriously, and not just from a, oh, what's wrong with you kind of perspective. And so I'm really, really glad to see real scientific inquiry taking place here. But, at the end of the day, all we ever want to do is just take a nice, warm, relaxing bath. Right? I don't know. Some people like baths more than other people. I like showers.
Will, if I have any say in the matter ever, all I ever take is baths. I could never take a shower again and just take baths. I'll be the happiest person in the world.
No,
sometimes
see, for
hours
me,
at a time.
See, I don't think I can relax enough to take baths. I sit and I'm like, okay, gotta get up and go places. I mean, I'd rather be in the shower, like moving around. Anyway, some people really like to go to baths. Some people go to Bath to take a bath, or to tour the ancient Roman baths. Why? I mean, and this I find very, this is just a thing. Why the baths in Bath? Why? I mean, is that?
It's the water. It's the
It is
weather.
It is. It is the water. No, really, truly. So the reason people would go to these baths, is a large part of it was not just to be social and be around other people. It's kind of like the the places where the waters are supposed to have healing powers. They're the tales of these Roman baths being healing and rejuvenating and helping people. And she's like, it's just a public bath. What are you talking about? It's just a spa, whatever. What's going on there?
Well, if the Roman soldiers didn't have any shower at home, it might just they didn't stink anymore. And that was healing to everyone around them.
Right. Well, there are questions that we need to answer, right? Is it just stinky soldiers needing a bath, or is it more than that? And researchers decided they'd sample the waters, the biofilm, and the sediment of the Roman baths. And they have just published their work in a, what is it called? The Microbe is the journal, Science Direct. It's open access. And their analyses of these samples from the baths in Bath, UK, have actually discovered a whole lot of biodiversity. So there's lots of bacteria, but not just archaebacteria that are in these baths. And bam, 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 antibacterial properties. So as we are on the hunt for new antibiotics, because, our antibiotics are starting to not work so well anymore, we're going to the natural source, bacteria who fight other bacteria, right? Or organisms, yeast, fungi, etc., who and archaebacteria who all are fighting with the bacteria. What do they have? What are the defenses they have? What's going on? So the water from the King's Spring in Bath was, it had the highest diversity overall, which is, interesting. But then in addition to discover that when they looked through metagenomic sequencing at the ribosomal RNA, did antimicrobial screening, they found that there were all sorts of different, like over 300 different bacteria, and that 15 of them had inhibition against pathogens, E. coli, Staphylococcus, Shigella, like things that are troublesome to humanity, to say the least. And there's the potential that this ancient Roman bath may not have just been the story of a healthy bathing environment that was rejuvenating and great, but actually may provide data and information for us moving forward to create are better defenses for us. I mean, I'm not telling you to go to Bath and jump in the baths. I don't think that's a great idea. But perhaps this information will give us new targets for our own antibacterial efforts as we try to fight these pathogens in our human existence. Pretty cool, right?
Yeah, and it really makes a lot of sense. It really makes a lot of sense that you would be having a large group of people share the same bath water, that the good microbes would win out over the bad because they are in greater abundance.
Yeah.
And so, yeah, fantastic. So from now on,
we should all bathe together. Social baths for health. Wait a minute. I don't think, no.
We used to do this.
It's in the water.
So we used to do this.
Yeah.
Back when the American family was, parents and 6 or 8, 9 kids, right?
Yeah.
The saying, "Don't throw the baby out with the bathwater," is because there was a hierarchy of who got to take the bath first. Mm-hmm. Usually started with Papa, then Mama, and then it went down in age by the kids. And the baby was last. Baby got the best biome of the bunch and the dirtiest water.
You hope. I mean, historically, babies were more likely to die. High childhood mortality in history. But anyway, we'll go past that. Beyond your argument. Okay.
but because in this day, Anyway, we got to the end of another episode of the This Week in Science the world's most popular science news podcast, YouTube live
Talk show, live broadcast.
version. Yeah.
Yes.
All right.
I think we've done it. I think we have gotten to the end of all the stories for this week. I hope everybody enjoyed the conversation and the new ideas or old ideas, but just made new because somebody looked at them again for the first time with data. Like, oh, look, if we look at it with scientific eyes, well, we're going to figure things out. I really hope you all enjoyed the show. And I do want to say thank you to those of you who help us make this happen. All of you in the chat rooms, thanks for being here and talking. Really good to see you. I see you in YouTube and Twitch and Facebook and Discord. I see you there. And I love seeing your comments. So I love seeing you interact with each other. Fada, thank you so much for your help with social media and show notes. Identity4, thank you for helping to record the show. Gord, Arnlore, others, Fada also, thank you for helping to make sure that our chat rooms are great, kind, thoughtful, curious places to be. And Rachel, thank you for editing the show. Additionally, thank you to our Patreon sponsors. And thank you to Justin's little cowlick for giving me eternal entertainment. Woo, I'm way down the list. Got to go to the top. Thank you, TWIS patrons. Thank you, Alan Viola, Aaron Anathema, Arthur Kepler, Craig Potts, Mary Gertz, Teresa Smith, Richard Badge, Bob Coles, Kent Northcote, George Corris, Pierre Velzar, John Ratnaswamy, Chris Wozniak, Vegard Shefstad, Jonathan Stiles, a.k.a. Don Stylo, Ali Coffin, Reagan Shubru, Sarah Forfar, Don Mundus, PIG, Stephen Alberon, Daryl Myshaks, Andrew Swanson, Fred S104, Sky Luke, Paul Runovich, Kevin Reardon, Noodles Jack, Brian Carrington, David Yee Youngblood, Sean Clarence Lamb, John McKee, Greg Riley, Mark Hessenflow, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Richard Brendan Minish, Johnny Gridley, Remy Day, G. Burton Lattimore, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul Philip Shane, Kurt Larson, Craig Glandon, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp. I see you in the chat room. Lawn Makes, I know you're here too. EO, Adam Mishkan, Aaron Luthen, Bob Calder, Marjorie, Paul D. Disney, David Silmerly, Patrick Pecoraro, and Tony Steele. Thank you so much for all of your support on Patreon. And if any of you out there want to support us on Patreon, head over to twist.org, click on the Patreon link, and choose your level of support for every month. Whatever you can give, we appreciate it. On next week's show,
we will be back Wednesday, 8 p.m. Pacific time, 5 a.m. Thursday, Central European time, broadcasting live from our Twitch or YouTube and our Facebook channels.
Oh, yeah. And if you want to listen to us as a podcast, we are a podcast. Just look for This Week in Science wherever podcasts are found, except for the Google Play Podcast Store, because now that doesn't exist anymore. And it's apparently YouTube podcast. I don't even know what to do. If you found the show, yeah. If you found us and enjoyed it, you got to get your friends to subscribe to, for sure.
Yeah, and then help them figure out where to find us next, because I guess I didn't even understand what Kiki was just saying. So for more information on anything you've heard here today, show notes, links to the stories will be available on our website, www.twist.org, and you can also sign up for newsletters. You can find amazing swag. We have swag that has sciencey implications that was made by sciencey hosts of the show, handcrafted, and then sent to a place to replicate on a pre-crafted item.
And you can sign up for our newsletter, because that'll come to you someday for sure. If you want to send us an email, let us know if you have feedback, a topic that you liked, if you didn't like, you want us to cover something, don't want us to, whatever, suggestion for someone that we should interview, you should, send us an email, put it in our social media accounts. Maybe, I don't know if we're going there anymore, but that email, if you send it, you got to put TWIS in the subject line, because otherwise it is going to end up getting sent into the middle of a Toxoplasma gondii parasite and stuck in somebody else's brain, and I don't know, going dormant, and then just like sitting there unless there's a virus, because then it'll like burn up in a fever. Just put TWIS in the subject line.
We look forward to discussing science with you again next week. If you've learned anything from the show, remember.
It's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.